Hi, I'm Jay Phelan. Let's talk about some biology. We're going to talk about here another way in which genetics doesn't work as simply in as straightforward a manner as Mendel would have us to believe. So, as I've reviewed previously, that when Mendel first talked about inheritance in pea plants, he talked about 7 different traits, but every trait was coded for by a single gene with 2 alleles, where one allele was dominant over the other allele, and there was no environmental effects, nothing was linked, and it made understanding what the results of his crosses were, making predictions and all that. It made it very straightforward. But in the real world, that's not always how it is. For many cases, genes are coded for by traits are coded for by a single gene, but not in every case. So we're going to continue building up a model of how genes influence the phenotypes of organisms. And what we're going to look at this time is the case of situations where a trait is coded for by a gene for which there's more than 2 alleles. Now, with an individual, no, there can never be more than 2 copies, right? Because we're a diploid species, so you have one copy from your mother, one copy from your father. So every gene's got 2 copies within an individual, but within a population, there can be lots of alleles. There can be just one allele, and for most genes in humans, that's the case. Probably for 75% of the genes, I'm estimating, there's just one. Everybody has 2 copies of it. Everybody has the same genotype. That's that. But there's still a large amount of our genome for which there are 3 or 4 or more than that. There can be dozens. There can be hundreds. For one eye color gene in fruit flies, there are about 1,000 different alleles that are present in the population. So we're going to investigate what happens when you have a gene that has more than 2 alleles. We'll look at the case of blood type in humans. People are kind of familiar with this. There are 3 different alleles possible for the gene that determines blood type. So we've got one that we call I with a superscript A. We have one with I with a superscript B, and one that's just I. And the way this works, it's sort of bizarre. I with a superscript A, we'll just call it the A allele. A is dominant to the lowercase I. So if you have both of those, then you're going to show that in the phenotype. B is also dominant to the lowercase I. If you have a I with a superscript B and a little I, you're going to be blood type B. But A and B are co-dominant to each other. If you have one allele of each, they're both going to show up in the phenotype. So for the population, 3 alleles. For any individual, just 2 alleles. This means that you can have people who are phenotype A, but they can be 2 different genotypes that they have. They can be homozygous for I with superscript A, or they can be A with a little I. In both cases, they're going to look like blood type A. Or we've got blood type B. Same thing. One phenotype, 2 different genotypes that give it because the B is dominant to the little I. Because A and B are co-dominant with each other, they're both going to show up in the phenotype. So if you have one allele of each, then your blood type is called AB. And then we have a 4th blood type, O. That's when you have 2 of the recessive allele, little I. So these are all the blood types that we see in a population, 4 different types, just from 3 alleles. Now, what's going on there? What does it mean? What do the alleles code for in blood type? They code for antigens, and these are chemicals on the surfaces of cells, and they're like a signpost saying, hey, I belong here. Here's what type of cell I am. I belong in this individual, so that the immune system can understand, all right, don't attack that cell. If you're blood type A, the A allele that you carry codes for an antigen that you put on all your red blood cells. It's a little thing, sticks out, and additionally, then that individual is going to produce antibodies, little immune system molecules that are going to attack antigens that don't belong in the body. It's going to produce antibodies for B. So I've drawn them in with silly little shapes. That the A antigen looks like a little triangle sticking out, and the B antibodies are like one of those blocks with a hollowed out curvy part. And then if you look at an individual that has the B allele, that codes for the B antigen. Well, what does the B antigen look like? It's the curvy little piece that sticks out like that. If you're blood type B then, and you carry the allele for the B antigen, your body produces A antibodies. So you can see the A antibodies, that would fit onto the A antigen. The immune system would attack that blood cell, get rid of it. The B antibody, with its curvy bit, that would latch onto the B antigen, and that would help the body to remove that foreign cell because it doesn't belong there. Then if you're type AB, you've got an allele coding for both different types of antigens. You put both of them on the surface of the blood cell, and you don't produce any of the antibodies. You can't, right? If you produced any of those antibodies, they'd attack your own cells, which you don't want them to do. So if you were type AB, no antibodies. And then blood type O, the opposite. You don't code for any antigens on the surface of the red blood cells, but you produce both different types of the antibodies, the A antibodies and the B antibodies. So you see you've got this whole different selection here. But if you look at this, then you can get insight into, what's going to happen if I put a bunch of AB blood cells into an individual that is blood type O? Well, they've got all those A and B antibodies. They're going to latch onto it. They're going to mount an immune response. The person's going to get sick. Bad news. On the other hand, if I put a bunch of O blood cells into someone, they have no antigens. Kind of a stealth blood cell. They go through, and regardless of which antibodies the recipient has, they're not going to be able to attack it because there's nothing sticking out. So this is why there are consequences for what person you can receive blood from and what person you can donate blood to. So I'm kind of giving away the answer here, but you should still think about this. People with blood type O are considered universal donors. I think of it as the O. That's what the cell looks like. It's just a clean, non-antigen-coded blood cell, so it can go anywhere. It's stealth. People with blood type AB are considered universal recipients. Why is that? You can figure that out. Here's a silly little problem. This wouldn't really happen, hopefully. 4 babies are mixed up in a hospital, we'll say, and we know the baby's blood type and the parents' blood types. We also know you have to figure out who belongs to whom. The whole point of this is just understanding that when you see a blood type, such as A, you don't know what the genotype is. They have an A allele and they have the little I, maybe, or they have 2 copies of the A allele. So if you go down this, you'd have to look for each baby, the AB baby, we understand, all right, there's one A allele, one B allele. So they'd have to have a parent that has the A and another parent who has the B, and so on. You go through here and you can figure it all out. Now, you'll have heard when talking about blood groups, not just that, I'm type O or I'm type A, but you'll hear O positive or AB negative. And there's another gene called the Rh blood group marker, and this also codes for different types of antigens on the surface of red blood cells. You don't just have the A and B. There can be all sorts of different markers on the outside of a cell. And what we have for the Rh group, there's just single gene, 2 alleles, and if you have 1 or 2 copies of the dominant allele, then you're called Rh positive. You produce those antigens on your cells. If you have 2 copies of the recessive version, then you don't produce any. You're described as Rh negative. So that also is going to be important when you are giving or getting a blood transfusion. So if you're Rh negative and you get Rh positive blood, then your immune system is going to attack those Rh antigens on the cells that you receive. And the reaction, the immune reaction, can vary from mild, you barely notice it, to severe, where this can even lead to death. So our take-home message here is that multiple alleles for a trait can cause the population to have many different phenotypes, but you have to always remember that each individual still only has 2 alleles, and the trait still follows Mendel's law of segregation in terms of how they're passing them on. So there it is, genes with more than 2 alleles.